Hello, everyone, and welcome to today's webinar from OpenEye Cadence Molecular Sciences. My name is Suhani Nagpal. I'm a lead application scientist here, and I'll be a host for this webinar. I'm delighted to introduce our presenter today, Dr. David LeBard, our head of target exploration at OpenEye CMS. And he'll be presenting on how you can own your protein target with TargetX. David has been with OpenEye over 8 years, brings extensive expertise in both theoretical and computational modeling of complex biological systems. His work focuses on building strong collaborations across industry and academia to address major challenges in drug discovery today. His research spans modeling passive permeability of drug-like molecules, rare event sampling of biomolecules, and more recently, cryptic pocket detection in proteins, areas that continue to expand our understanding of molecule behavior and therapeutic design. David is also well known for delivering quite engaging and highly informative talks. So we are in for an insightful session today. Before we get started, a couple of housekeeping items. We welcome questions throughout the talk. Please enter your questions in the chat window on the right, and we'll address those toward the end. If we run out of time and cannot get to everything, we'll follow up with you in the coming few days via email. And with that, I'll hand it over to you, David, on owning your target with TargetX.
Thanks, Suhani, and thank you all for joining me today. I'm really happy to talk to you about kind of a follow-up to a webinar I had last year, which was on drugging the undruggable. And here we're really digging into trying to solve undruggability through our new product called TargetX. And so the title of my talk is Own Your Target with TargetX, Expand Pocket Space, Prioritize by Ligandability, and De-risk drug discovery decisions. And I hope by the end of this talk, I've convinced you that TargetX can help you with those three things. So the talk will proceed as follows. We'll start out by explaining what is so hard about this problem. And really, it boils down to the druggable genome just being too small and how TargetX can help you expand it. From there, I'm going to tell you about what we had to do to build TargetX, which was we had to build our own data set of pockets. And this new data set is called Groovy, and I'm going to talk about that and compare it to other pocket data sets that are out there. We took Groovy and we built a ligandability model that we call TargetX, and I'll explain how we did that. And then we went back and we went through our entire cryptic pocket detection workflow, and we wanted to validate it. Now that we had a strong model for ligandability, we built a second data set that we're calling Encore that allows anyone to validate cryptic pocket workflows. So we took Encore and we applied our TargetX workflow and we validated our workflow using Encore. So that's the talk. I'll leave you with some conclusions and outlook once that's over. So let's go ahead and start. The druggable genome is just too small, and let me show you how we can expand it. So to give you an idea about the druggable genome, I think everyone was kind of educated by a paper that came out about 25 years ago that's called the druggable genome. And this is really what it said. It said, given a human being, and in this particular case, since it's ski season here in Santa Fe, I thought I would give a shout out to the Trevian skier here, who is basically the perfect human being. But this human being has about 30,000 genes in their genome, which corresponds to about 30,000 proteins. Now, if you ask the question, how many of those proteins are actually involved in the pathways of disease, that's about 10% of those proteins. So about 3,000 proteins of the 30,000 are actually associated with disease. So this is the full landscape of what pharma could actually go after if they want to, make medicines that actually cure diseases. Now, unfortunately, what this paper showed is that only 2 to 5% of those disease-associated proteins are actually susceptible to traditional orally available rule of 5 drugs. So on the upper end, that's about 50 proteins. On the lower end, about 60, or 150 to 60. So there's really not that many. If you look at that difference between the amount of disease-associated proteins, 3,000, to the ones that are susceptible to typical rule of 5 molecules, there's a massive gap in there. And the question is, how can we bridge the gap between those proteins we know are involved in disease and those that are susceptible to drug-like molecules. So we want to expand this druggable genome, but how can we possibly do it? And the way I'd like to convince you that we can do this is by validating and investigating targets by using the TargetX workflow. So we have a TargetX workflow that we believe can help expand this druggable genome far beyond where it stands today. And how we do this are three separate, really challenging calculations. One, we have to do some enhanced protein sampling. And we need to do this because we have to make our protein visit rare states that it wouldn't normally visit in a brute force simulation. And we do this by combining two techniques together. First, we have normal mode driven weighted ensemble molecular dynamics, where we project a set of normal modes and we follow those projections through our resampling weighted ensemble procedure. And then this allows us to sample a huge amount of protein phase space. Now, combined with that, we also run these simulations in the presence of a xenon co-solvent. And this allows us to not only open proteins that require some conformational selection process from the protein, which would be driven by the normal modes, but also those pockets that may need some encouragement to open through an induced fit process. And that's where the xenon comes into play. So that's our enhanced protein sampling. And of course, I've talked about this in webinars before. Now, there's the second equally challenging problem, which is once you have this massive ensemble of trajectories, how do you comb through them to find the pockets that may represent some place that you could put a drug-like molecule? And we have two methods that we use. One, which is the expose-on method that looks at correlated solvent exposure, and a related method to this expose-on method, which is our dynamic probe binding method, which looks at high occupancy binding sites. Now, our pocket detection methods, like all of them, frankly, have one serious downfall in that they often predict too many pockets. And if you have too many pockets on a protein, then it really makes starting a structure-based drug design program fairly challenging. So what you need on top of that is a way to rank those pockets. And that's why we've developed this TargetX ligandability model to help you prioritize those pockets and perhaps de-risk your system and fully understand it from a target exploration perspective. So the talk will proceed as follows. I'm going to tell you about how we built the TargetX model, and then I'm going to go backwards and say, well, with that TargetX model, can we validate the full workflow of enhanced protein sampling and pocket detection using the pocket ranking from TargetX? So I've told you about the druggable genome and how TargetX can expand it. Now I'm going to tell you how we built TargetX. And first, what we had to do was build a suitable data set for training models such as TargetX. So if you're familiar with the field of ligandability modeling, there are many popular data sets out there for both training models and validating models. Now, if you look at what those training sets actually contain, it's kind of striking and unfortunate. So here, what we've done, we've taken the two most popular data sets for training ligandability models, which are the SC-PDB dataset and the Chen11 dataset, shown respectively on the left and right hand panels. And what we've done, we've taken the molecules out of the data set and we've done clustering based on their Tanimoto combo. And I'm showing you here the highest occupancy clusters that we find. And we've colored them based on their chemical nature. And what you see is somewhat unfortunate. So the SCP-PDB is almost entirely composed of nucleotides in the top 13 out of 15 clusters. On the other hand, Chen11 contains many nucleotides, but also completely biased towards carbohydrates and many excipients. Now, unfortunately, we don't have a massive amount of drug-like molecules that are actually represented here. So what we're training on are pockets that are primarily binding nucleotides and carbohydrates, which unfortunately is a vast amount of proteins across the human genome. So if you're trying to find or make a model that is able to predict ligandability for drug-like molecules, these may not be suitable. Now, if we look further at the validation data sets that people use once they've built their model to validate whether the model is suitable or not, this is the case. So we did the same type of clustering based on the Tanimoto combo, using all of our toolkit knowledge from our OpenEye toolkits. And what we find again is that both of the validation data sets from Holo 4K, shown on the left bottom, and the Coach 420, shown on the right bottom, they're both highly biased towards nucleotides and carbohydrates. So there's extreme bias in molecules that are not really representing drug-like molecules. Therefore, the pockets you would not expect to necessarily bind the types of molecules that we tend to develop in the pharmaceutical industry. Now, one other really unfortunate aspect of this is that if you look at the training set and the validation sets, they all represent the same types of molecules, which means they have the same type of pockets, which implies that there's strong data leakage between the training sets and the validation sets. Now, what this will mean is that if you build a model based on the training data from SCPDB or Chen11, and then you validate it on Holo 4K or Coach 420, you're likely going to have elevated success rates that are not true simply because the data has leaked from the training to validation. So what we need to do is build a non-redundant data set that better describes drug-like molecule pockets rather than pockets for nucleotides, carbohydrates, or excipients. And that's where our new data set called Groovy fits in. So we've tried to take the PDB and boil it down to just those structures that have pockets that represent where drug-like molecules actually bind. And this is how we built the Groovy dataset. So we started out with an input structure of about 200 protein crystal structures taken from the RCSB. And these were all in Orion, on our Orion platform inside MMDS. And then our first filtering step, we just extracted all the PDB structures as design units. So we threw away any duplicate design units and anything that couldn't be prepared by Spruce, and just retained those structures as fully prepared design units. Then from those design units, we looked at the Iridium score, which is based on the overlap between the experimental X-ray density and what the atomic model actually says. And there's a score which is called Iridium, which was developed at OpenEye a few years ago. And we only kept those high trustworthy scores, the most high quality scores, out of those extracted PDB design units. From those structures, we further filtered them down to only structures that are bound to drug-like ligands. So anything that was an excipient or perhaps a nucleic acid or anything else like that, we discarded because we only wanted structures with pockets that represent where real drug-like molecules bind. The next filtering step was deduplicating any pockets with near identical ligands that may be nearby. So oftentimes you have a situation where two or more ligands can bind to the same protein in really similar parts of a pocket, and we wanted to completely remove, the double or triple occupancy problem from that. And finally, we further removed any pockets with high pocket similarity based on a shape and color, which is a pseudo-electrostatic term based on our site hopper algorithm. And from these 200,000 protein structures, after this filtering, we were left with about 1,847 structures that we call the Groovy dataset. Now, the Groovy dataset has further been split on an 80-20 split to training, which we call Groovy training, and then a 20%, which we call Groovy validation. And our plan right now is to publicly release the Groovy validation dataset so anyone can test their model on these type of pockets. So I've told you about the druggable genome, about pocket datasets, the bias that's contained in them, and how we tried to remove the bias and data leakage from our Groovy dataset. And now I'm going to tell you about how we built a new ligandability model called TargetX that is built on this Groovy dataset. So we use the Groovy training portion of the Groovy dataset to build the TargetX model. And how did we do that? Well, first we had to build a feature vector. And we did this using entirely API calls that are from our toolkits. And from that, we have a set of feature vectors that we think are important, including the potential at a given point on the surface, the curvature of the surface, the solvent exposed atom count and atom density, the partial charge at that particular point, the aliphatic atom count or aromatic atom count, the hydrogen bond acceptor or donor count, the carbon, nitrogen, or oxygen atom count. And then we use this feature vector and we evaluated this feature vector on a set of surface points that are decorated on a protein surface. And using this evaluation of a surface point, we've trained TargetX on a balanced amount of positive and negative control pockets from Groovy training. And in previous webinars, I've explained how we did this separation to provide both a positive control pocket, which is a known pocket, and a negative control, which is a random part of a surface. And then we evaluated it on the pockets known from Groovy training, and we trained it and then validated it on our Groovy validation dataset. And this is how the validation of the TargetX model looks. And we try to validate this based on what's commonly used in Bayesian statistics, which is known as a Bayes factor. And a Bayes factor tells you the probability of given two hypotheses, which one is more likely. So in this particular case, you have the ratio of the probability of the normalized ligandability score giving a hypothesis that the pocket is actually ligandable over the probability of the normalized ligandability score being non-ligandable. And the larger this number, the more evidence you have that something is ligandable versus non-ligandable. So if we take the Groovy training set and we apply it to the set of known pockets and the negative controls, this is what we see. We see a clear separation between the known pockets that are shown in red and the negative controls that are shown in gray. And you'll notice that the normalized ligandability score goes from 0 to 1. And using the Bayes factor, we were able to determine a cutoff for ligandable and non-ligandable based on this typical Bayes factor classification for the strength of evidence for the what we're calling ligandability. And you can see here that 0.27 is where you get the cutoff between non-ligandable shown in gray and the known pockets that are shown in red. And the strength of evidence that you see at the bottom of this plot shows that you have marginal evidence at the cutoff, which increases from substantial through strong all the way to decisive as the ligandability score increases. And on the right-hand side, I'm just showing you just the raw brute force performance of this model, looking at both the true positive rate and the false positive rate. And we see that the area under the raw curve is 0.97. So we feel like this is a strong model. But this is not fully validated yet against what we would consider to be known drug-like molecule pockets. So we did a further validation using known FDA-approved binding sites, and we used this to construct a set of false negatives and true positives. And we chose the true positives to be those that are greater than our cutoff at 0.27, and the false negatives to be less than 0.27. But of course, all of these are representing FDA-approved drug binding sites. And the performance of this model from this data shows that we have a high precision, 90.97, and a high accuracy, over 90%. And we made two different flavors or architectures of this model, both a Gaussian process and a random forest regression model. The random forest regression model showed slight improvement over the GPR model, so that's the one that we use by default, but we provide both of them on Orion today. So with that, we're fairly happy that we've made a model that can distinguish real drug-like molecule binding sites from those that do not bind drug-like molecules with high accuracy and high precision. And we're going to use this to further validate the workflow for the TargetX validation. So to do this, we had to build a second data set, as I told you earlier, that we're calling Encore, that allows you to really validate the soup to nuts workflow, starting from an APO structure all the way to a predicted pocket. Can you not only open up a pocket, but also predict its presence and tell me that that pocket is one that I should start with for my structure-based drug design? So to build the Encore dataset, we thought hard about the types of pockets that you typically see that bind drug-like molecules. And the 6 categories of pockets that we've defined are the following. One pocket, which involves simple side chain motion, where a single side chain needs to get out of the way of a pocket to open it up. A slightly more complicated version of this is a loop that has to open up to expose a pocket. Alternatively, the loop may be hanging out somewhere else and needs to close back on top, and we call these reverse type pockets. So there are times when you may have a forward or reverse pocket that could appear. Similarly, secondary structure can change, and you can move it completely out of the way to form a pocket, or fold it in, similar to the loop motion on top, to form a pocket. There's also a secondary structure change that may happen, where you may go from some order to disordered state, or the reverse process, where you go from an order to disorder, but folding a pocket on top. There's also interdomain motion, where 2 domains of a protein, which may or may not be tethered, may need to pull away to expose a pocket or collapse together to form the pocket. And finally, there's a 6th type of pocket that's not necessarily cryptic, in that it already exists in the protein, but you still have to be able to detect it. And this is a pre-existing deep pocket. And these pockets are a pocket without really any known solvent channel. So we took these types of pockets and built a data set using this categorization to try and validate our workflow. And this data set is called Encore, and we're going to make this publicly available in an upcoming publication that allow anyone to detect or to validate their pocket detection workflow. So Encore provides both holo and apo structures of the same protein, so you can see how far you have to move to go from the closed state to the open state, and also it will allow you to validate that you're doing a good job when you are opening it. So this is what the Encore dataset looks like. From those 6 different pocket types that we listed here, we have 21 different proteins and 26 different pockets that are represented from these proteins. And again, they're drawn from the pre-existing pocket, sidechain motion, loop motion, secondary structure motion, secondary structure change, or interdomain motion. And we've further subclassified certain changes within these pocket openings for characteristics that we're calling order to disorder changes, which may be required for certain pockets to open, but not others. And if we look at the contents of this data set, we can categorize it based on the Guide to Pharmacology categorization of proteins. And in the top right-hand pie chart, you see that most of the proteins are enzymes. We have quite a few kinases that are represented, some ion channels, and then other protein targets make up the second largest class. And of these pockets, the majority of them are allosteric, but there are also some orthosteric pockets as well. So this is the Encore data set that will be publicly released soon, that we're using now to validate our cryptic pocket detection workflow that we're calling TargetX. So let's validate TargetX using this Encore dataset. So this is the performance of TargetX on the Encore dataset that I just showed you. And this is a fairly busy slide, so I'm going to walk you through it from top to bottom. So here I'm showing again the same categorization that we had of the 6 pocket types, but I'm showing you here the ability to our ability to predict the presence of those pockets using our full workflow. So in the top area of the plot, you'll see a pocket RMSD plot as a function of the different pockets across the different proteins. And we've also shown you here whether they're orthosteric or allosteric underneath those. Now, to understand the plot, what I'm showing you here in pink are the RMSD differences between the apo and holo pairs of the Encore dataset from just the crystal structures. And you can see that those apo-holo pairs can be quite high from one another. So this is not just full atom RMSD, but rather just the RMSD of those heavy atoms inside the pocket residues themselves. And you can see some of the pockets are upwards of almost 8 angstroms away from each other. So really large scale of changes. In purple, you'll see what we were able to predict as that pocket from our weighted ensemble simulations. And the RMSD is based on the holo-like frame and the apo structure from the Encore dataset. And what you'll notice is, in many cases, the purple is much lower than the pink, which suggests that our simulation protocol is dramatically opening up the pockets and forming a pocket that's much closer to the apo state than it is to the holo state. Now, we tried to further characterize whether or not we're fully characterizing the holo state by measuring the pocket RMSD to the holo state structure that's in the Encore dataset. And this is shown in orange, magenta, and green, where orange is less than 1 angstrom, magenta is less than 2 angstroms, and green is greater than 2 angstroms. And you can see that the majority of these, for the holo-likenness, we're actually getting quite a few that are less than 2 angstroms. So not only are we able to predict the pocket, but we're also able to predict a holo-like pocket that you would expect a drug-like molecule to bind. Now, finally, we're taking those pockets and we're trying to rank them based on their ligandability. And we've used a null model, which is F pocket, versus our actual ligandability model, which is target X. And I'm showing this in the very last section on the bottom here in both black and blue. Blue represents a failed prediction. Black is a successful prediction. And you can see that F pocket definitely fails to predict pockets where large-scale changes are required. That's because F pocket doesn't do anything. It's only operating on the apo state. However, our predictions from our holo-like pockets, we're able to use target X, and it predicts a ligandable pocket for all but 2 different pocket types in the same protein. It was both, in this particular case, Hep C virus polymerase, and this, we missed a pre-existing pocket and a pocket that required some order to disorder transition. Both of these are under investigation right now at OpenEye, and so hopefully next time I give a webinar, I'll be able to better tell you why we were not able to sample that. But this is how it looks today, and I'm fairly convinced that this solution, because it's able to predict the pocket over 90% of the time, is the most predictive method for pocket holo-likeness and ligandability that I've seen before in the literature. So we're really proud of it. But I'm going to focus on one example here, which isn't exactly the best, but I think is fairly typical of when we are successful. And this is the case of biotin carboxylase, which is right here in the interdomain motion, where F pocket failed. We were able to get the holo-likeness fairly well into angstroms, but not perfectly. And I would just want to highlight that. So just focusing in on biotin carboxylase, this is what we see. So in the right-hand side, you see an apo structure, which is taken from the crystal structure from the Encore dataset. And the RMSD to the holo state, which is shown in gray, the apo structure is shown in green. The differences there are pretty striking. So you can see in the figure in the top right that not only do side chains have to move, there's a tyrosine up on a loop there that you can see has to have a dramatic movement, but there's also secondary structure elements that need to move as well. And using our normal mode-driven weighted ensemble, you can see the weighted ensemble frame in blue on the left-hand side compared to the holo-like state again in the gray. And this is fairly striking. We were able to move that tyrosine so it's able to make strong interactions with the ligand. The secondary structure elements move out of the way. We were able to actually predict the holo-like state. Now, that's not the end of the story. We're not able to do this with every structure, so this is an area of continued research, but I think this is the direction we're trying to push towards, which is, can we actually predict not only a pocket, but the most holo-like pocket, which would bind a drug-like molecule? So I've told you about the druggable genome, how it's too small, and how TargetX can expand it. I told you about how pocket data sets are biased and how data leakage occurs between training and validation, and how we built a new data set called Groovy that we hope eliminates those problems for the field. We've also built a new model based on Groovy training set, and we've validated it, and it is highly accurate and highly specific. We've also further built a new validation data set for pocket detection workflows that we're calling Encore. We've validated against Encore and showing that the TargetX methodology works over 90% of the time. So now I'd like to leave you with some conclusions and outlook. First, I'd like to say the druggable genome is just simply too small. And if we want to cure all human disease, we need to go beyond these rule of 5 susceptible sites and be able to prioritize proteins and targets based on ligandable pockets. And we've done this at OpenEye by making a new method called TargetX that combines enhanced protein sampling with a xenon-based mixed solvent that also has pocket detection and ranking for a full end-to-end workflow that will help you not only prioritize pockets, but will help you de-risk and make better decisions for structure-based drug design programs. Finally, as I said, dataset bias is real. It exists in models that are trained and validated for ligandability, and we've made a new dataset called Groovy that we believe addresses that. And finally, we've made another dataset for validation called Encore. The Encore dataset we have validated against our TargetX full workflow. And although we do have some failures when it comes to pre-existing pockets and order to disorder, overall, the results are incredibly tantalizing, and we're really proud of the results. And all of this work is under peer review right now, and there should be 2 publications coming out in the next few months that describes this in full detail. And with that, I will go ahead and turn it back to Sokhani, who will be happy to answer or take any of your questions.
Thank you, David. That was an insightful talk indeed. We have a few questions coming in, so let's move into the Q&A portion of the webinar. I'll start with the first one, and this is about AI folded structures. How do you see the interplay of AI-based structure prediction with weighted ensemble MD-based pocket discovery? So could predicted structure be used as starting points?
That's an interesting question that I think we and everyone else have thought a lot about. And we've done something towards answering that. I don't think we have the full answer yet. So we did the first thing, which is asking the question, can an AI structure fold it to the holo-like state? You know, is our workflow even necessary in the first place? And so to answer that question, we took our Encore model and we folded it using the sequence, and we measured the RMSD of the pocket and asked whether it's closer to the apo state and the holo state. And on average, it was closer to the apo state than the holo state. So it suggests that even if you have a AI folded structure, AI predicted structure, it isn't necessarily going to give you the whole like pocket. Now, once we were able to do that, we actually turned those into design units. Now, once you have a design unit inside Orion, it can be used anywhere across our workflows, including in the TargetX area. So we know that it's useful, but I can't tell you how well it would perform on those because we haven't performed the experiments ourselves.
I see. Okay. And do we have something to facilitate that on Orion?
Yeah, we do actually. So we have not just the Boltz 2 flows, but also OpenFold and many other popular open prediction models are being incorporated into Orion all the time. But those are the two that we have available today.
Great. The next one is about experimental data. So, could any experimental information be incorporated into the TargetX workflows that you showcased for a more experimentally guided cryptic pocket discovery?
Yeah, so what I showed here was driving weighted ensemble simulations using the normal modes that are encoded in the protein structure. But that's not exactly the same thing as using experimentally derived constraints. And so we have a method to use either cryo-EM maps or X-ray densities as a way to kind of drive your system to new conformational states. And we've been able to showcase this already with our cryo-EM work, where we're able to take a structure that's pretty far away from a cryo-EM map, drive it to that map, and then use all of that trajectory data and still search for pockets and rank them. And we were pretty happy to show that for one particular system, HER2, we're able to open up a pocket where a known antibody binds and correctly predict that rank as the number one ranked pocket as well. So those are the ways that we can incorporate experiments. We would love to work with partners that have maybe HDX data or NMR data to open up the toolbox for experimentally driven simulations a little bit more. But today, that's what we're limited to.
That's quite promising to hear. The next question is about how well does the target X model generalize to cryptic pockets that were not part of the original training data set?
Yeah, so we've looked at that fairly extensively. And although our model performs well on many different pocket types, there's a few that we know it doesn't perform well on because we removed those examples from the training data, or there were not enough examples to begin with. And those include peptides, peptidomimetics, macrocycles, and we also purposefully removed covalent inhibitors, although we have some evidence that our model still can predict ligandability for those covalent sites. So even though it wasn't trained on it, it tends to generalize across those. But we know that it would be really beneficial to us to have a larger set of data to kind of incorporate some of those beyond the 5 molecules as part of our ligandability model. But we probably need a partner for that, and it's not something that the public data has to offer.
Yeah, I understand. And it'll definitely expand the scope of our model. Yeah.
Yep. And we really hope to.
Okay. The next one is about the Encore dataset in terms of its structural diversity. Which classes of proteins kind of benefited the most from the TargetX workflow? Have you tested this on, let's say, membrane proteins?
So yeah, the TargetX workflow, as I said, it's primarily composed of soluble proteins. There is a cytosolic domain of a transmembrane protein in that data set, but it's not the transmembrane portion. Um, but we are working to expand that, and I have ongoing research in my group where we're actually actively pursuing a target GLP-1 receptor, where we're showing that we can take the same workflow that we developed for these soluble proteins and detect and rank pockets correctly on fairly large transmembrane proteins. So that work is underway and hopefully comes out sometime by mini cups in the fall.
Excited to hear about it. Could you elaborate on the estimate of computational cost per protein?
Yeah, so because Orion is a platform that is really made to kind of do everything for you, one thing we like people to kind of remove from their minds is the costs. And really, I want people to think about the time. And so, and that I mean the actual wall clock time. So the TargetX workflow, as it stands today, you spruce prep a protein and then you click a button and you wait about 24 to 48 hours. And in the process, the TargetX workflow does all the protein sampling, does the pocket detection, and the ligandability ranking, and then provides you that output at the end of the day. So if you think about it in that perspective, each target is about a day to 2 days before going from an apo structure to a predicted pocket with a whole likeness that we think is pretty high. But the other thing to consider is that in the process of that protein sampling, we're doing an enormous amount of simulations, and we actually generate between 3 to 5 microseconds of simulation time per simulation, which represents a huge amount of data for all of us MD folks out there. So we're really happy that you can provide a large amount of data with about 1 to 2 days worth of compute time.
That's quite reassuring to know. So it sounds like there's an automated pipeline. You just click a button and you would get candidate pockets at the end of the pipeline.
That's exactly right.
Okay, that's great. Well, I think in the interest of time, we'll follow up on the remaining questions via email. David, thank you for the great talk. And with that, I'll bring the webinar to close. For all our attendees, I would like to thank you all for your time and your participation. We look forward to seeing you at the next webinar from OpenEye Cadence Molecular Sciences. Until then, goodbye.